You know, when we think of a supervillain, we usually picture like someone with this grand destructive plan.
Right, like a comic book bad guy trying to blow up the moon or something.
Exactly. Or, at the very least, someone loud, someone making this massive entrance and demanding everyone's attention.
Yeah, totally.
But if you think about it, the most successful villains, the ones that actually come back year after year and always get away with it, their main superpower isn't brute strength. It's the ability to constantly change their disguise. Like, you never even know who you're looking at until it's way too late.
Which honestly perfectly describes a threat that you, the listener, probably encounter every single winter.
Yep.
It's not a comic book villain, obviously, but a microscopic one. The influenza virus.
Right. So welcome to today's deep dive. Our mission today is to really look under the hood of what we just casually call the flu.
And we've got a lot to cover.
We really do. We pulled together this fascinating stack of virology journals, epidemiological reports, and CDC historical archives to unpack what this virus actually is.
Because it's not just chicken soup and tissues.
No, definitely not. We're going to explore how it constantly shapeshifts, the devastating history it carries, and, why epidemiologists are monitoring it literally right this second.
Yeah, the stakes are very real.
But to understand how this shape-shifting villain works, I feel like we first have to look at how we even identified it.
Right.
Because for centuries, humanity had absolutely no idea what it was fighting. Like, zero clue.
Oh, yeah. It took hundreds of years and some very bizarre historical detours just to put a name to it. I mean, we can start with the word itself, right? Influenza.
Which I have to say sounds almost, well, elegant in a way.
It does.
But looking at the historical archives, the origin is wild. It actually comes from medieval Italian astrology.
Yeah, that always blows my mind.
Right. So between the 14th and 16th centuries, Italians blamed these periodic sudden epidemics of, coughing and fevers on the influence of the stars.
Right. It was literally the influenza of the cosmos.
Right.
Or sometimes they blamed the influence of cold, damp air.
Wow.
And the name just stuck permanently after this major epidemic swept through Europe in 1743.
That is just crazy. But that astrological origin kind of highlights a fundamental problem, doesn't it?
How so?
Well, for a very long time, the flu was defined entirely by its clinical picture. Like you had a fever, chills, body aches, what doctors called a flu-like illness.
Right, because no one could actually see the pathogen.
Exactly. They just categorized it by how it made you feel. But, okay, let's unpack this because this feels like a crucial turning point in the sources. There was this major shift in 1933 where the definition changed from a clinical one to a virological one, meaning scientists started identifying it by its genome sequence, its surface proteins, its host range in a lab.
Which changed everything. But honestly, I want to push back on this a little. Why does this shift actually matter to, the average person? If you feel sick with a fever and body aches, doesn't sick just mean sick?
Wow.
Like, why does the CDC care so much about laboratory confirmation over your actual symptoms?
That is a great question. Because that clinical to virological shift, it exposed something deeply uncomfortable for the medical field. Yeah, it turned out that many illnesses that doctors confidently called influenza, based on a patient's symptoms, they were not influenza at all.
Wait, really?
Yeah, they were entirely different respiratory viruses that just happened to cause similar fevers and aches.
So doctors were treating the wrong villain entirely. They were, like, fighting imposters.
Exactly. And it goes both ways, too. Real virologically confirmed influenza infections are sometimes completely mild or even asymptomatic.
Yeah.
So if public health officials only went by symptoms, they would miss a massive portion of the infected population entirely.
Right, that makes sense. You can't track how a pathogen spreads if your tracking method is based purely on who happens to feel terrible on a given Tuesday.
Right, right.
Today, defining influenza by laboratory confirmation, like PCR testing, it's the only way to know what invisible enemy is actually circulating.
Okay, so how did we actually capture it in a lab? Because the discovery story in these archives is just wild.
It really is.
It wasn't just some lone genius looking through a microscope, right?
Yeah.
After the horrific 1918 pandemic, scientists spent over a decade hunting for a bacterium.
Yeah, they thought the flu was caused by a bacteria called Haemophilus influenza.
Which is such a mouthful.
Yeah.
And they were completely on the wrong track. That name is basically just a historical artifact of a massive error.
Exactly. The actual breakthrough required pivoting to viruses, which are vastly smaller than bacteria.
Right.
So in the early 1930s, researchers, specifically Richard Shope in 1931, managed to isolate a filterable agent, a virus, causing flu-like illness in pigs.
Okay.
But isolating the human version remained notoriously difficult until a British research team in 1933, Smith, Andrews, and Laidlaw, tried using ferrets as an experimental model.
And here's where it gets really interesting for me. How did they even know ferrets were susceptible to human flu strains?
Well, this is the best part.
It's so crazy. A lab assistant who happened to be sick with the flu literally just sneezed near the experimental ferrets, and the ferrets caught it.
It's perfect. It's a perfect example of how scientific breakthroughs often rely on just a bit of accidental, absurd luck.
Seriously.
That human-to-ferret sneeze established a crucial animal model. It turns out ferrets have respiratory systems that respond to the flu very similarly to ours.
Oh.
So that accident finally gave scientists a way to culture and study human influenza in the laboratory.
What's wild, though, is that the boundaries of what we even consider the flu are still moving today.
Oh, absolutely.
Like just back in 2011, researchers found a completely new type influenza D circulating in cattle.
Right, out in the agricultural world?
Yeah, it primarily infects cattle and pigs, and it has no confirmed role in human disease. Virologists are actually debating if it should even be conceptually grouped with the human relevant types.
It's kind of like finding a distant cousin of our supervillain who just hangs out on farms and doesn't bother anyone.
That is exactly it. But when we talk about the ones that do bother us, we're almost always talking about influenza A.
Yes, that is the big one.
That is the type responsible for seasonal epidemics and every known global pandemic. And to understand why we can't just, cure it once and for all like we did with polio or smallpox, we really have to look closely at its biology.
We do. We have to open the Shapeshifters playbook.
Right. Let's get into the mechanics here. So, influenza A is characterized by two specific proteins that sit on the surface of the virus.
Right, the H and the N.
Exactly. Hemagglutinin, or H, which currently has 18 known versions, and neuraminidase, or N, which has 11 versions. That is where we get names like H1N1 or H3N2.
Think of those two proteins as the virus's toolkit. The hemagglutinin in the H is the key the virus uses to unlock and enter your healthy cells.
Okay, the key.
Right. And the neuraminidase, the N, is like a machete it uses to cut newly replicated viruses free from the dying cells so they can go infect others.
That's a great visual.
And our immune system trains itself to recognize the specific shape of that H key and that N machete. But influenza's genome makes that incredibly difficult.
Because of how it's structured.
Exactly. Most viruses carry one continuous strand of genetic material. Influenza A carries a genome broken into 8 separate, distinct pieces of RNA.
8 separate pieces. It's like a disassembled piece of furniture packed in a box.
That's a really good way to put it. And that fragmentation is the root of nearly everything unusual and dangerous about the flu.
Right.
It enables 2 distinct processes of viral change: antigenic drift and antigenic shift.
Okay, let me try an analogy here to separate these two, because this feels like the absolute core of our deep dive today.
Go for it.
So antigenic drift is the slow process, right? The virus relies on an enzyme called RNA polymerase to copy its genetic code. But this enzyme is incredibly sloppy.
Extremely sloppy.
It doesn't have a backspace key to fix typos, so it makes constant tiny copying errors.
Right.
It's like the virus is slowly changing its disguise over time. First it puts on glasses. A few months later it grows a mustache.
I love this.
And eventually the immune system's facial recognition,
Yeah.
Your antibodies from last year's infection, it just slowly stops working.
Yeah.
Because it doesn't recognize this guy in the glasses and mustache.
That is a perfect way to visualize it. Accumulated over months and years, these small point mutations subtly alter the physical shape of those H and N surface proteins.
Okay.
The key still opens your cells, but it looks different enough to evade prior immunity.
Which is deeply relevant to all of us. I mean, when you walk into a pharmacy to get your annual flu shot, you are literally getting the result of scientists trying to outguess this antigenic drift.
Exactly.
They are trying to predict what disguise the virus will be wearing 6 months from now.
Right.
That's what causes the seasonal flu.
Right.
But then we have antigenic shift,
Yeah.
which is, well, it's far more terrifying.
Shift is the sudden, rare process. It is entirely different from drift.
Shift is like, okay, the virus walks into a phone booth with a completely different virus,
Right.
and they swap their entire bodies out in one go.
Right.
It steps out instantly, wearing a high-tech stealth suit, totally unrecognizable. It's sudden, and it means there is zero pre-existing immunity in the general human population.
And this brings us back to that disassembled furniture genome. Shift happens specifically because of those 8 separate gene segments.
Right, the separate pieces.
If 2 different flu strains infect the exact same cell simultaneously, their segments can literally mix and match as new virus particles are being assembled.
Wow.
It's a wholesale swap of an entire gene segment. This reassortment produces a hybrid with totally new surface proteins.
But wait. If a bird flu and a human flu need to swap genes, they have to physically infect the exact same host cell at the exact same time.
Yep.
But human viruses prefer human receptors, and bird viruses prefer bird receptors. How do they even meet? They need a biological middleman, which is why we always hear about swine flu, right? Pigs have both.
You've hit on the exact virological mechanism. To infect a cell, a virus has to bind to a specific receptor.
Okay.
Humans have what's called alpha-2-oral-6 sialic acid receptors, which line our upper respiratory tract.
Got it.
Avian influenza viruses, or bird flu, preferentially bind to alpha-2-oral-3 receptors, which are common in bird intestines and in the deep human lung, actually.
So they shouldn't easily mix, normally.
Normally, no, but pigs are one of the few animals whose respiratory tract carries abundant amounts of both receptor types.
The mixing vessel.
Exactly. So a pig can inhale an avian strain from, say, bird droppings, and a human strain from a farmer at the exact same time. Both viruses enter the pig's cellular phone booth, their 8 segments shuffle together, and a novel, highly contagious hybrid virus spills back over into the human population.
That is terrifying.
The 2009 H1N1 pandemic was a perfect example. It was a quadruple reassortment carrying gene segments traceable to North American birds, human seasonal lineages, and 2 separate swine lineages.
And that exact mechanism of antigenic shift is what creates catastrophic pandemics. Which brings us to the most notorious outbreak in human history.
Yeah.
An event that forced scientists to completely rethink how this virus actually kills people.
The 1918 pandemic.
It killed an estimated 50 to 100 million people worldwide. That is more deaths than World War I itself. That number is just staggering.
It's hard to even wrap your head around.
And what's wild is who it killed. Normally, seasonal flu has a U-shaped mortality curve, right? It mostly kills the very young, whose immune systems are naive, and the very old, whose immune systems are weakening. So the 2 peaks of the U.
Exactly.
But 1918 had a W-shaped mortality curve. There was a massive abnormal spike right in the middle among healthy adults aged 20 to 40.
An age group that is normally the absolute most resistant to respiratory viruses. For decades, that W-shaped curve was a complete medical mystery.
Right.
But the leading explanation today is that this specific 1918 strain provoked a cytokine storm.
Wait, let me make sure I'm getting this. A cytokine storm is an immune overreaction, right?
Yes, exactly.
So if it killed healthy young adults because of a cytokine storm, does that mean having a strong, robust immune system was actually a fatal disadvantage in 1918?
That is the tragic paradox of that pandemic, yes.
Bro.
Cytokines are basically signaling proteins that tell your immune system to send troops to an infection site. But the 1918 virus triggered such a massive, unregulated inflammatory response that a vigorous immune system became the mechanism of harm.
Oh my God.
The body sent so many immune cells and fluid into the lungs to fight the virus that the patients essentially drowned from the inside out. Their own robust defense systems caused severe tissue damage.
That is just awful. But wait, how do we even know the exact mechanism of a virus that disappeared 100 years ago?
We reconstructed it.
After the fact.
Right.
And the story of how they did it in the sources is just straight out of a thriller.
It really shows incredible scientific persistence. So in 1997, a retired pathologist named Johann Holton traveled to a remote village in Alaska.
Okay.
He exhumed the body of a woman who had died in 1918 and was buried deep in the permafrost. The permafrost acted like a natural freezer, perfectly preserving her lung tissue.
And the crazy part is, Holton had actually attempted this exact same recovery back in 1951 as a graduate student.
He did, yeah.
He dug through the ice back then, but the technology just didn't exist yet to amplify degraded genetic material. So he failed, waited nearly 50 years, and goes back in his 70s to try again.
It's amazing. And this time, PCR technology had caught up. He sent that frozen tissue to molecular pathologist Jeffrey Taubenberger.
Right.
Now, extracting fragile, degraded RNA from an 80-year-old frozen lung is incredibly difficult. But Taubenberger's team managed to sequence the fragments and laboriously piece that extinct 8-segment genome back together by 2005.
That is unbelievable.
It's one of the only times scientists have fully rebuilt an extinct pandemic pathogen from the remains of its victims.
So if our immune systems can overreact that violently, it brings up another massive question about how our bodies remember the flu. And this leads to some intense controversies. Let's look at a concept called original antigenic sin, first described by immunologist Thomas Francis Jr. in 1960.
This is a foundational debate in immunology.
Yeah, this is the debate over whether your very first childhood exposure to the flu permanently biases your immune system for the rest of your life.
Right. The mechanism works like this. Your immune system has a deep memory. The idea of original antigenic sin is that your immune system remembers the very first flu strain you caught as a toddler so fiercely that when it encounters a related but mutated strain later in life, it mounts a defense against the old strain instead of adapting to the new one.
Oh, wow.
It essentially fights the ghost of flu past rather than the reality of flu present.
Even when you get vaccinated against the newer strain.
Exactly. The immune system is stubborn. It prefers to deploy its existing memory rather than learn a new trick.
That is fascinating.
And some researchers argue this explains why certain age cohorts survive specific pandemics while others perish. Like during the 2009 H1N1 pandemic, older adults actually showed unexpected partial protection.
Really? Why?
Because their childhood exposure to earlier H1N1-like strains back in the 1950s gave them a lingering biased immunity that actually helped them.
Okay, so if we know our immune systems are incredibly picky and subject to this memory bias, it puts an immense amount of pressure on how we design and manufacture vaccines.
Oh, definitely.
Which brings up a massive bottleneck, the egg adaptation controversy.
Right.
I mean, why on earth are we dealing with a virus that rapidly shapeshifts? And our primary way to fight it is a manufacturing method from the 1940s involving injecting it into chicken eggs.
You're hitting on the exact bottleneck that frustrates modern virologists. Growing a human-adapted virus in fertilized chicken eggs is cheap and scalable, which is why we do it.
Right.
But viruses adapt to their environments. When you force a human flu virus to grow inside a bird egg, it faces selective pressure. It naturally mutates its hemagglutinin surface protein to bind better to avian receptors so it can survive in the egg.
Wait a minute. So the act of manufacturing the vaccine accidentally forces the virus to mutate again?
Unfortunately, yes.
You're basically training the human immune system to recognize a disguise the virus isn't actually wearing in the real world?
That is the exact problem. Several flu seasons in the 2010s saw vastly reduced vaccine effectiveness, and researchers attribute it partly to this egg adaptation.
That is so frustrating.
The vaccine virus had drifted away from the circulating virus just by being grown in eggs. This is fueling an urgent, ongoing push toward cell-based and recombinant vaccine production methods that avoid the egg passage step entirely.
And the pressure to get these vaccines right can lead to sheer panic and public health missteps. Like, take the 1976 swine flu vaccine incident.
Oh yeah, a classic case study.
A soldier died at Fort Dix from a novel H1N1 strain. Health officials looked at the data, feared a W-shaped mortality curve repeat of 1918, and launched a massive campaign, vaccinating over 40 million Americans in months.
But the virus never achieved sustained transmission beyond Fort Dix.
Right.
The pandemic never materialized. And worse, the vaccine was linked to an elevated incidence of Guillain-Barré syndrome.
Let me quickly explain Guillain-Barré for anyone unfamiliar, because the mechanism is terrifying.
Please do.
It's an autoimmune reaction. The immune system gets confused by the vaccine, and instead of just attacking the virus, it starts attacking the myelin sheath, the protective coating around the body's own nerve cells. It causes muscle weakness and sometimes paralysis.
It is a rare complication, but because they vaccinated 40 million people, those rare cases piled up visibly.
Yeah, people noticed.
The program was abruptly halted. It remains a textbook case study in the immense tension between precautionary public health action, trying to stop a pandemic before it starts, and the catastrophic fallout of a false alarm.
And speaking of anticipating pandemics, the controversy doesn't stop at vaccines. It extends into the laboratory with the highly pathogenic avian H5N1 gain-of-function debate.
This is a huge, huge debate right now.
Yeah. So H5N1 bird flu has a fatality rate in humans far higher than seasonal flu, but thankfully it has never achieved sustained human-to-human transmission.
Right.
But in 2011 and 2012, two research teams, Fouchier and Kawaka, published studies showing they had engineered H5N1 to become airborne among mammals.
They did this by passaging the virus through ferrets. Essentially, they infected a ferret, took the mutated virus from its lungs, infected another ferret, and repeated the process until the virus naturally evolved the ability to transmit through the air.
Wow.
This is what we call gain of function, forcing a pathogen to acquire a new ability in a lab setting to understand how easily it could happen in nature.
But they essentially proved this deadly bird flu could become a highly contagious human pandemic, which ignited a fierce global biosecurity debate.
Absolutely.
I mean, should the genetic sequence that makes it airborne be published openly where bioterrorists could see it? Should this research be conducted at all?
And that single debate deeply shapes how viral research is regulated today, because the line between understanding an enemy and accidentally arming it is incredibly thin.
Which is why it is so crucial that we clear the air on some of the rumors and misconceptions swirling around the flu today. Let's do some rapid-fire myth-busting
Let's do it.
and explain why all this biology matters to you right now. First myth. The Spanish flu didn't start in Spain.
There is zero evidence it started there. Candidate origin sites actually include military camps in Kansas or transit hubs in France.
Okay, so why Spain?
While during World War I, combatant nations heavily censored their news to protect wartime morale, Spain was neutral and had a free press.
Ah, I see.
They reported the devastating outbreak extensively, so the world falsely assumed Spain was the epicenter. It's a stark reminder that even the naming of a pathogen can be shaped by politics and censorship rather than biology.
Right. Next myth. The stomach flu is influenza. If you are vomiting for 24 hours, do you have the flu?
Absolutely not. Influenza is strictly a respiratory virus. It attacks your airways and lungs.
So what are people catching then?
What people colloquially call the stomach flu, which causes vomiting and diarrhea, is usually caused by entirely different gastrointestinal pathogens like norovirus or rotavirus. The shared name is just a persistent public misunderstanding.
Good to know. What about the classic excuse everyone hears, I'm not getting the flu shot because it gave me the flu last time?
Right, we hear this all the time. The standard injectable flu vaccine contains an inactivated, dead virus, or just synthetic pieces of the virus.
So it's literally dead.
Yes, it biologically cannot replicate or cause an infection. The soreness, mild fever, or fatigue you might feel for a day is just your immune system waking up and building defenses. It's a sign the vaccine is working, not a sign of infection.
But what about the nasal spray?
Even the nasal spray vaccine, which is live, is cleverly engineered. It is attenuated or weakened so that it can only survive in the cooler temperatures of your nasal passages.
Oh, that's smart.
It literally cannot survive in the warmer environment deep in your lungs where real influenza does its damage.
Okay, last myth.
Okay.
Let's relate this back to the listener's expectations of modern medicine. People see a bad flu season and think the scientists messed up the vaccine formulation.
Right.
But it's really a real-time evolutionary race, right? The vaccine effectiveness is often 40 to 60%, specifically because the virus is actively drifting in the 6 months after the strain is selected.
That is the harsh reality of viral speed. The WHO's Global Influenza Surveillance and Response System tracks strains worldwide and selects the vaccine formula roughly 6 months before flu season.
To allow for manufacturing.
Exactly, for manufacturing and distribution. But 6 months is an eternity for an RNA virus. A season with lower vaccine effectiveness usually reflects the virus mutating away from the predicted strain, not a failure of the vaccine concept itself.
So what does this all mean for us today? The WHO estimates seasonal influenza causes hundreds of thousands of respiratory deaths globally every year.
Yeah, a steady, tragic toll that rarely makes headlines.
But the stakes regarding a new pandemic are very real right now. H5N1 avian flu is currently causing large-scale outbreaks in wild birds, poultry, and notably, U.S. dairy cattle.
With sporadic human infections in farm workers, yeah.
And this is why those surface proteins matter so much. Epidemiologists are currently watching the hemagglutinin receptor binding sites on this dairy cattle virus, like hawks.
They are searching the genome for any specific mutations that would signal a shift toward alpha-206 human receptors.
Right. Understanding drift and shift is not historical trivia. It is the exact framework scientists use right now to assess pandemic risk in real time.
So, to bring this all together, the flu isn't just some winter inconvenience. It's a segmented, shape-shifting RNA virus.
Yep.
It relies on the slow, disguised change of antigenic drift to ruin our winters, and the sudden phone booth body swap of antigenic shift to threaten the globe.
Well said.
We combat it through global surveillance, but we are always one pig or one reassortment event away from a massive new challenge.
It is a relentless, microscopic, evolutionary arms race.
It really is. I want to leave you with a final thought to mull over, one that could change the future of this arms race entirely. We've talked extensively about how the flu relies on mutating its disguise, the head of those hemagglutinin and surface proteins, to evade our immune system.
Right.
But what happens if we target the parts of the virus that never change?
Oh, you're talking about the stalk of the protein.
Exactly. Imagine the surface protein is a mushroom. The cap of the mushroom is constantly changing colors and shapes to confuse us.
But the stalk holding it up remains structurally identical year after year. Scientists are currently trying to build a universal flu vaccine that trains your immune system to ignore the changing cap and exclusively attack the stalk.
Which would be incredible.
A single shot that could theoretically end the shape-shifting arms race forever. We might just be one scientific breakthrough away from retiring this supervillain for good.
Let's hope so.
Thank you for joining us on this deep dive. Keep questioning what you think you know.